Today we are going to explain immune response mechanisms part 1, B and T lymphocytes. First, B lymphocytes. We will study the origin of B lymphocytes, the biology of B lymphocytes, and the clonal selection theory. The origin of B lymphocytes. B lymphocytes and its secreted end product, the antibody molecule, has been recognized as a powerful immunologic tool for over a century. Studies in birds showed that the bursa of Fabricus, a hindgut lymphoid organ, was the site of early development of antibody-producing cells. This lineage of cells was therefore termed B cells. In mammals, cells of B lineage are initially generated in the fetal liver when hematopoietic stem cells migrate to the liver from yolk sac. This process begins during the 8th week of human gestation. The fetal liver continues to be a major site for production of the erythroid myeloid series, including B cells, until well into the second trimester. Stem cells then populate the bone marrow. Thereafter, B cells are continuously produced in the bone marrow throughout life. B cells develop from a pluripotential stem cell that can give rise to all of the different types of hematopoietic cell types. Biology of B lymphocytes. B lymphocytes display immunoglobulins as integral proteins of their cell membranes. These membrane-bound immunoglobulins are the B cell receptors for antigen, and they differ from secreted immunoglobulins in several ways. They are anchored in the cell membrane with the carboxy terminal embedded into the membrane. The transmembrane region is hydrophobic in nature. The amino or antigen binding end is extracellular and strategically placed to receive foreign antigen. Receptors differs from immature B cells and activated B cells. In immature B cells, it acquires the immunoglobulin M monomers on its surface. And as B cell developed proceeds, immunoglobulin D molecules appear on their surface membranes along with immunoglobulin M, and immunoglobulin D becomes the predominant membrane-bound isotype found. C3D, Epstein-Barr virus, and C3b and Fc portions of immunoglobulin G molecules, histocompatibility class 1 antigens. All these receptors are present on the surface of immature B cells. When B cells become activated, increase the amount of HLA-D region encoded molecules appear on its surface. Receptors for Fc, gamma, and C3b also show an increase, then decrease during the terminal stages of B cell differentiation. Activated B cells also display receptors for interleukin 2 and other factors needed for full differentiation of B cells. We should note here that immunoglobulin D molecules are not found on newly formed B cells. Immature B cells respond negatively to cross-linkage of their immunoglobulin M molecules by multivalent antigen. Thus, early exposure to antigen eliminates B cell responsiveness, leading to tolerance toward that antigen. Here, the receptor molecules on the B cell surface. We here find immunoglobulins, interleukin 2, Fc receptors, C3b and C4, Epstein-Barr virus, and lipopolysaccharide. Also, T cell growth factors appear. After antigen or mitogen stimulation, B cells can proceed along one of two pathways. They can differentiate into plasma cells and secrete large amount of immunoglobulin, or they can divide and return to a resting stage. If they differentiate into plasma cells are the terminally differentiated state of B lymphocytes. They release thousands of antibody molecules every second. The plasma cell seldom divides and has an average lifespan of less than 4 days. If they divide and return to a resting stage, these cells are called memory cells and can rapidly differentiate into plasma cells following second exposure to the same antigen. The clonal selection theory. How do cells make antibodies in such enormous variety? One antibody can neutralize just one type of antigen, and the antigens come in an incredible diversity of shapes, sizes, and chemical compositions. Yet the human system is capable of manufacturing a practically unlimited range of antibodies against bacteria and other toxins, viruses, pollen grains, incompatible blood cells, and even some novel man-made molecules. The clonal selection theory of antibody formation. The conceptual father of the clonal selection theory was Paul Ehrlich. Over 100 years ago, he postulated that a white blood cell's surface bore receptors with side chains to which foreign substances became chemically linked. This building prompted the cell to produce and secrete copies of this bound receptor, the antibody, into the circulation. Later, Niels Jern, who introduced the concept of clonality and formulated notion similar to Ehrlich's side chain theory, he suggested that any animal possesses in its armamentarium small numbers of performed antibodies against all antigens. An antigen-antibody complex interacts with white cells and more antibody to the same antigen is produced. Later, David Talmage in 1957 went a bit further to say that white cells are selected for proliferation when the antibodies they synthesize matches the invading antigen. In retrospect, it is obvious that Jern and Talmage laid the foundations of the clonal selection theory. It remained for Macfarlane Burnett to crystallize the concept of clonal selection. Central to Burnett's thinking was the tenet that one cell produced just one kind of antibody. He proposed that binding of an antigen with an antibody receptor on the cell surface triggers the cell to multiply and manufacture more of the same receptor. He asserted that each cell and its clones or offspring can produce just one kind of antibody, and he coined the term clonal selection to describe his theory. Here, the clonal selection theory, according to Burnett, antigen selects a perfect fit from a library of preformed receptors. Here the antigen. Then the B cells become activated. It began to proliferate. Some cells become plasma cells and begin antibody synthesis. Some cells became memory cells to memorize this type of antigen for the second attack. The human system is endowed with antibody-producing cells bearing an entire library of antigen-binding receptors, the immunoglobulin molecule. This occurs during development. Each B lymphocyte becomes genetically programmed through a process called gene translocation to make a unique B cell receptor. Molecules of that B cell receptor are placed on its surface where it can react with epitopes of an antigen. If an antigen enters the system, it matches with one of the receptors and binds to it. This induces the cell to proliferate to produce several clones of its kind, each capable of secreting more of just than one particular type of receptor or antibody, matched for the invading antigen. Once an entire cadre of cells has been generated, the second response to the same antigen is more intense, since there are many more cells awaiting to bind the antigen. Each cell will then generate more clones secreting the same antibody. Once clones of antibody producing cells have been generated, prolonged exposure to antigen is not necessary to maintain antibody production. The clonal selection theory, together with an understanding of the genetics of antibody diversity, has provided many answers regarding the mechanisms which control the production of vast repertoire of antibodies that the immune system is endowed with. Finally, clonal selection explained immunologic tolerance as the complete deletion of an entire clone of cells, which would occur before or after birth if an antigen overwhelmed the metabolic capabilities of the cells. In 1960, Macfarlane Burnett shared the Nobel Prize with Peter Madwer for his accomplishment in understanding immunologic tolerance. In 1984, Niels Jerne also received the Nobel Prize for his theoretical contributions in conceptualizing the idea of clonality in immunology. Second, we have the T lymphocytes. We will study the origin of T lymphocytes, molecules on T cell surface. There are two main types of T lymphocytes, T4 lymphocytes or CD4 positive cells, and T8 lymphocytes or CD8 positive cells or cytotoxic T lymphocytes. Then we will study the T cell receptor for antigen and another type known as regulatory or suppressor T cells. T lymphocytes mediate two general types of immunologic functions, effector and regulator. The effector functions are mediated by 2 points. The first, secretion of soluble substances called lymphokines or cytokines. Second, by their ability to kill other cells by cytotoxicity. The regulatory functions are represented by their ability to amplify cell-mediated cytotoxicity by other T cells and by the help they render in the production of immunoglobulins by B cells. These functions also require the synthesis of lymphokines or cytokines. Origin of T lymphocytes. The thymus-derived lymphocytes or T cells it appear as stem cells that migrate to the thymus and move from cortex to medulla and out into the periphery, a journey that takes 3 days. As T cells gain maturity, certain surface molecules begin to be expressed. An understanding of T cell function has been possible because of the identification and the characterization of these surface molecules. Here we have molecules on the T cell surface. We have the T cell subsets, T helper or T4 cells, and the T cytotoxic cell or T8 cells. T helper cell or T4 cells, main cell surface markers are CD4, CD3, T cell receptor, and CD2. Restrictions, MHC class II. Functions, stimulate B cell to produce antibody, induce CD8 T cell cytokine secretion, and macrophage activation. The second subsets are T cytotoxic cell or T8 cell. Main cell surface markers are CD8 positive, CD3, T cell receptor, and CD2. Restrictions are MHC class I. Functions, lyse antigen bearing target cells. Stem cells do not display any surface molecules. As T cells move into the thymic cortex, the CD2 molecule is the first to appear, followed by CD3, CD4, and CD8. In the thymic medulla, two distinct lineages become evident, one bearing CD2, CD3, and CD4 molecules, and the other bearing CD2, CD3, and CD8 molecules. In peripheral lymphoid tissue, 65% of the cells possesses CD2, CD3, CD4 molecules on their surface, and 35% display CD2, CD3, and CD8 antigens. CD4 positive T cells or T4 cells constitute the helper T cells, and CD8 positive T cells or T8 cells form the cytotoxic or suppressor T cells. In the thymus, all T cells learn to recognize self-MHC gene products, and this helps them to react with antigen in conjunction with MHC gene products in the periphery. T4 lymphocytes or T4 helper cells or CD4 positive cells. Functionally, there are different types of T4 lymphocytes based on the cytokines they produced. The two primary types are T helper 1 cells and T helper 2 cells. T helper 1 cells are lymphocytes that recognize antigens presented by macrophages and function primarily to activate and heighten cell-mediated immunity. T helper 2 lymphocytes recognize antigens presented by B lymphocytes and function primarily to activate and heighten humoral immunity. Here we will study T helper 1 cells. We said that T helper 1 lymphocytes recognize antigens presented by macrophages and function primarily to activate and heighten cell-mediated immunity by producing cytokines such as interleukin 2, tumor necrosis factor beta, and interferon gamma. Collectively, these cytokines lead to enable T8 lymphocytes to proliferate and differentiate into cytotoxic T lymphocytes capable of destroying infected host cells and mutant cells. To activate cytotoxic T lymphocytes and natural killer cells, activate macrophages, enabling them to destroy intracellular pathogens. 3. Stimulate the production of opsonizing and complement activating antibodies for enhanced attachment during phagocytosis. 4. Activate neutrophils, stimulate increased production of monocytes in bone marrow. 5. Function as chemoattractants for phagocytes. T helper 2 lymphocytes. T helper 2 lymphocytes recognize antigens presented by B lymphocytes and function primarily to activate and heighten humoral immunity by producing cytokines such as interleukin 2, 4, 5, 10, and 13. Collectively, these cytokines lead to enable activated B lymphocytes to proliferate, stimulate activated B lymphocytes to synthesize and secrete antibodies, promote the differentiation of B lymphocytes into antibody-secreting plasma cells, and enable antibody-producing cells to switch the class of antibodies being produced. Enable B lymphocytes to activate eosinophils and produce increased amounts of immunoglobulin E against helminths. Immunoglobulin E acts as an opsonizing antibody, enabling eosinophils to attach to the helminths for extracellular killing. T8 lymphocytes or CD8 cells or cytotoxic T lymphocytes. Cytotoxic T cells constitute one of the body's major defenses against viruses, intracellular bacteria, and tumor cells. These cytotoxic T cells are effector cells derived from T8 lymphocytes during cell-mediated immunity. However, in order to become cytotoxic, naive T8 lymphocytes must become activated by cytokines produced by antigen-presenting cells. This interaction between antigen-presenting cells and naive T8 lymphocytes occurs primarily in the lymph nodes, the lymph nodules, and the spleen. Now we will study the T cell receptor for antigen. The T cell receptor for antigen. T cells must have a receptor capable of recognizing antigens. The T cell receptor is a heterodimer composed of two chains, each of molecular weight 40 to 50,000 Daltons, linked by a disulfide bond on the T cell surface. The two chains are termed alpha and beta chains and are integral membrane proteins extending 4 to 5 amino acids into the cytoplasm. Each chain is folded into two domains, one having a relatively constant structure, much like the constant region of the immunoglobulin molecule. The other domain exhibits far more variability than the immunoglobulin molecules and is analogous to the immunoglobulin variable region. It stands to reason, therefore, that the variable regions are strategically placed and constructed to bind with foreign antigen. Here, the T cell receptor for antigen, we can see the alpha and beta chains, we can see the disulfide bonds, and the constant regions and the variable regions. And we can see the CD3 molecules that is usually combined with the T cell receptor. Genetic organization of the T cell receptor. A study of genetic organization of T cell receptor led to the understanding that T cells too are capable of recognizing an enormous range of antigens. Separate sets of genes encode the alpha and beta chains. The genetic locus containing the alpha chain genes is present on chromosome number 14, while the genes coding for the beta chains are present on the 7th chromosome. These genetic loci can be further divided into regions similar to the gene arrangement for the immunoglobulin molecule. The regions have been named the variable region genes V, diversity segment genes D, joining region J, and constant regions C. Each gene region is comprised of gene clusters. For example, there are 60 V region genes for the alpha chain and 21 for the beta chain. Other regions also are highly polymorphic and have several gene clusters. The polymorphic genetic organization of T cell receptor. Here we can see the V region. It has 60 genes for alpha chains and 21 for beta chain. D region has 2 for beta chain. J region has 40 for alpha chain and 12 for beta chain. C region 1 for alpha chain and 2 for beta chain. The polymorphic genetic organization of the T cell receptor rearrangement occurs between gene segments analogous to that described for the immunoglobulin molecule. This provides the diversity to account for the existence of over 1 million distinct T cell clones per individual, each having a different antigen specificity. Antigen diversity is accounted for not only by rearrangement of gene segments as with the immunoglobulin molecule, but also by interactions between alpha and beta chains of the T cell receptor molecule. Therefore, different antigen specifications are created when a single alpha chain interacts with different beta chains. The CD3 complex. In all immunocompetent T cells, the T cell antigen receptor is non-covalently but intimately linked with closely related molecules called the CD3 molecule complex. The three peptide molecules of the CD3 complex have been called CD3 gamma and epsilon and theta. Each of the CD3 molecules is an integral part of the T cell membrane and extends into the cytoplasm further than either the alpha or beta chains of the receptor. The CD3 molecules transduce the antigen recognition signal received by the alpha and beta heterodimer to the inside of the cell. Surface CD4 and CD8 molecules. The CD4 and CD8 molecules play important role in T cell activation. Both the CD4 and CD8 molecules are the genes that encode them have been identified. There is no evidence of polymorphism or gene rearrangement in the case of these molecules, and they appear identical in cells having different antigen specifications. Therefore, they do not seem to participate in the antigen recognition. The CD4 and CD8 molecules have been shown to function as recognition markers for the MHC gene products. The presence of CD4 on T cell indicates that the T cell is programmed to recognize and interact with cells bearing MHC class II molecules, while T cells bearing CD8 molecules recognize cells displaying MHC class I molecules as part of their antigen specificity. It has been hypothesized that the CD4 and CD8 molecules are not involved in activation of T cells, but can increase interaction between T cells and target cells. They seem to behave like extra glue so that activation can occur. For cells seeing antigen for the first time, this glue may play an important role. It is also thought that since they are structurally constant, they play a part in recognizing the constant regions of the MHC class I and II molecules. This is borne out by the finding that the CD4 and CD8 molecules are closely associated with the T cell receptor and actually bind to the constant region of the MHC class I and II molecules, thus guiding the T cell receptors onto the antigen. CD2 molecule. The CD2 molecule on T lymphocytes is a transmembrane surface glycoprotein which facilitates cell-cell contact. The amino terminal domain of CD2, domain 1, mediates its adhesion function by binding to LFA3 or CD58, another cell surface glycoprotein widely expressed on various cell types including hematopoietic and epithelial cells. Both CD2 and CD58 are members of the immunoglobulin gene superfamily. The importance of CD2 function in the normal human immune response has been well documented. First, recognition involving helper T cells and antigen presenting cells. Second, for the cytotoxic effector function of natural killer cells and the cytotoxic T lymphocytes. The immunoglobulin gene superfamily. All the molecules involved in antigen recognition described so far are members of what is known as gene superfamily. There are the heavy and light chains of the immunoglobulin molecule, the alpha and beta chains of the T cell receptor, the alpha and beta 2 microglobulin chain of the MHC class I antigen, and the alpha and beta chains of the MHC class II antigen. It is obvious that all of these molecules have certain common characteristics. They all recognize and bind foreign antigen, and to do this, they are equipped with variable regions as part of their structures. This is consequent to the polymorphic gene structure and characteristic gene rearrangements seen in all of these molecules. It has been found that certain sequences about 110 amino acids long are common to all three structures. Last, we have regulatory or suppressor T cells. Regulatory or suppressor T cells, the existence of specific suppressor T cells and the phenomenon of immunologic suppression has fallen into disfavor in the present decade. However, most immunologists believe that some form of suppression exists. The reason for the suppressor cell controversy is that no one has been able to clone a unique suppressor T cell. Of the original suppressor T cells, the most promising recent candidates have been given other names. Examples of these are TR cells, TR1 cells, TH3 cells. TR cells. Most CD4 T cells belong to either T helper 1 or T helper 2 subsets. However, some from 5 to 10% of them do not. These have been termed T regulatory cells. Their surface receptors, they express a transmembrane protein called CD25, which is the alpha chain of the receptor for interleukin 2. Like other T cells, they also express the alpha beta chains, the T cell receptor for antigen, and can only be activated if it binds to the peptide class 2 MHC molecule. Their function: if activated, they secrete large amounts of interleukin 10 and often some transforming growth factor beta as well. Both these lymphokines are powerful immunosuppressants, inhibiting both T helper 1 helper cell mediated immunity, including graft versus host disease and inflammation, and T helper 2 helper antibody production, and possibly the action of CD8 cytolytic T lymphocytes, that is to protect the body against autoimmunity. TR1 cells. TR1 cells resemble TR cells in several ways. Although they do not express large amounts of CD25 on their surface, they require interleukin 10 for their formation and once mature, secrete large amount of it. TR1 cells are abundant in the intestine, and their chief function may be to make the host tolerant to many antigens that are part of its diet. It is believed that patients who develop Crohn's disease lack the down regulation characterized by interleukin 10 via the TR1 cells, making them intolerant to a number of dietary and other antigens. T helper 3 cells. TH3 cells are also prevalent in the intestine, but unlike TR1 cells, their main lymphokine is TGF beta. Also, like TR1 cells, they suppress immune responses to ingested antigens. Further research is needed to elaborate the relationships between these and other T cells that suppress immune responses. This work is relevant for patients who risk rejection of organ transplant and for the management of patients with autoimmune disorders like lupus erythematosus and insulin-dependent diabetes mellitus and Crohn's disease. Thank you.